So as a therapeutic strategy, we, and as I said, Dan and Jim separately have published with Jen Chow the idea that perhaps things can be shuttled towards the mitochondria, and if they're shuttled towards the mitochondria, less will get to the ER and therefore be secreted, and that would lead to fewer deposits. At the same time, we'd be enhancing mitochondrial metabolism, which would potentially then decrease the amount of theoretically glucose that could be consumed by the RPE, allowing it to pass through the photoreceptors and providing energy for the outer retinal system. So those were all reasons that we're interested in pursuing this general hypothesis. In order to pursue it, we first have to remember that the reticulosuttodrusen and drusen both contain apolipoprotein E, and ApoE is the most abundant apolipoprotein in drusen, although there are definitely other components in drusen. And as I said, in reticulosuttodrusen, it's only ApoE. So we've been using, besides actual fats being secreted, we've been using ApoE as a proxy for what the RPE is, the types of fats that the RPE is secreting, admitting that that's a definite assumption and that needs to be more proven. We're doing a lot of our studies in RPE cultures in which we grow them on transwells, which I'm sure everyone is familiar with. That allows maximal polarization of the RPE and also allows us to collect supernatants from both the apical and the basolateral side to understand separately what's being secreted into each compartment. We then will give an intervention that we think enhances lipid degradation, and we'll measure indeed whether lipid is being degraded, lipid degradation is being enhanced, and at the same time we'll collect supernatants from the basal and apical side, and we'll measure ApoE, as well as we're starting to measure both by enzymatic kits and by mass, eventually mass spectrometry and thin layer chromatography, actual lipid species that are being secreted into the media on each side. So that's the general platform and the hypothesis that the lab, globally wants to pursue. To get just a little bit more into the experimental setup, because a lot of the data I'm going to show you is going to rely on really understanding how we're measuring beta oxidation. We're doing it by proxy, which is that we're adding a fatty acid into our culture, and then we're looking for whether we see an enhancement or a spike in oxygen consumption rate, which would suggest mitochondrial beta oxidation is happening. So we use this device, and I think some are now familiar with this device, but others probably not. This device is called Recipher. And it depends on the fact that in culture, at least, all the oxygen is coming from above the media. And so the oxygen forms a diffusion gradient all the way down to the cells. The cells are consuming oxygen, so the oxygen tension is lowest down here. The oxygen tension is, of course, highest up at the media surface. And this device will run an oxygen sensing probe up and down in a precise way in the media itself, not touching the cells, measuring the oxygen gradient. If the cells are consuming a lot of oxygen, then the gradient is quite steep. If the cells are consuming very little oxygen, then the gradient is quite shallow. The advantage of this device is that it can run in our standard cell culture plates. In fact, we can even do this on our transwells and measure other aspects of cell health. This can be done over the course of weeks and even more than a month of continuous OCAR readings. So you can manipulate the culture, understand how it responds, then see how it recovers, then manipulate the culture again and see how it responds. So you get a lot of interesting data out of this type of experiment. And just as an example, and I presented this last year, so I'm not going to belabor the point, but when you put different levels of media on the cells, these are the exact same cells, cultured exactly the same way, you can see that the oxygen consumption rates are much lower when there's 200 microliters of media than when there's 65 microliters of media on the cells. And that's because at 200 microliters, the oxygen availability way up high is so far away from the cells that you've actually induced hypoxia at the level of the cells. And so there's just a maximum amount of oxygen that can be consumed. Essentially, the cultures are extracting all of the oxygen that's available to it and is therefore living in a hypoxic environment. So I presented this data last year, but I just want to re-emphasize it because it means when I'm doing all these experiments or the lab's doing experiments, we have to be very careful that we're using media volumes that are not limiting in terms of O2 consumption, especially because we're measuring processes like beta oxidation that have very high oxygen consumption rates to them, or should have high oxygen consumption rates to them. So in all the experiments that I'm presenting here, we've done all the controls to make sure that we're not in oxygen limiting conditions and the media volumes are appropriate.
Jason, can I just ask a quick question here?
Sure.
So in a previous slide, you showed your setup and you said you're using RPE cells. Are these human RPE cells? Are they primary cultures? Are they iPSC-derived RPE cells? What is the nature of your starting material?
Thank you, Steve. Yeah, I don't know if I should put your hairs on edge by telling that we're using undifferentiated fibroblastic ARP19, or tell you the truth that we're using primary human RP cultures that are well differentiated, have been in culture for at least 8 weeks. And also IPS RPE cultures. And on the primary human, we both grow adult and fetal. So we have all 3 cultures around available to us.
Great, thank you.
So the last thing for background on the experimental setup for everyone to realize is that just by playing around with this device, we have been able to know that as OCAR is established in the culture, wherever the OCAR is before an intervention, if the intervention is just a media change, that will predict where the OCAR will be after the media change. So this black line just represents standard media. We then change the media. It takes a while for the O2 gradient to reestablish. So this device does not measure things in short OCAR in short amounts of times. You have to let a new oxygen gradient reestablish in order to get an accurate OCAR reading. But as you can see, it plateaus back out, and it's at the exact same OCAR reading that it was at before. In contrast, I'm not telling you what the intervention is here, but this purple line was at a baseline here. We did an intervention, and then we clearly see the OCAR is much higher after the intervention. This triggered an OCAR spike. So in the data I'm going to present to you, what we tend to do is we take the average OCAR value over some period after the intervention, and we subtract it from the OCAR value before the intervention, and we are presenting to you delta OCARs. And that's what I mean when you'll see the y-axes on the graphs that I'm going to show you. That's what I mean by the delta OCAR. What increase in OCAR did we get on account of the intervention?
And how do you select the range of times over which to take that average?
We make sure to do it so that we fit our data to exactly what we want it to look like. I'm sorry. Yeah, so that's a
no. I agree. We all try to make our data as biased as possible. Please continue.
Yeah, no. So that's exactly right. We need to be very consistent, sort of just like when you're trying to do microscopy and quantification. You have to be unbiased in where you're sampling, et cetera. So we know that fatty acids have a certain sort of lifespan in our cultures. We'll take that sort of time period after the intervention, and because the gradient takes a while to establish, we try to catch it right as it looks like an equilibrium is starting to happen. So that, that's why, for example, the window started here on the left side.
Jason, sorry.
Sorry. Go ahead, Daniel.
Okay, I'll just continue this because it's on the same topic. Is there any sort of intent to take an area under the curve kind of measurement, which could be used to translate, for example, exactly how much oxygen has been consumed due to fatty acids specifically, and then you could even maybe use this sort of formula to determine how much carbon has been consumed. Or
yes, Dan, as you astutely pointed out, if you basically just took this curve here and just moved it back over here, you would get an area under the curve, which would represent, how much total oxygen was consumed, using whatever the intervention was. In this case, a known concentration of fatty acids at a known volume, in the media. And so you're exactly right that you can actually, calculate total, moles of oxygen consumed, which theoretically could give you total amount of the fatty acid that was shunted into beta oxidation. But it's a lot of us, it's a mathematical calculation that you think you can trust, but I'm taking it sort of not as hard fact.
Kind of along the same lines that Daniel was asking, why not taking the peak of oxygen consumption and subtracting that from, because that
Right.
would give you at least a precise value rather than putting an artificial window.
Right. The reason is as it will become clear as we go along, which is that the dynamics of different fatty acids and different interventions change in, even here, I'm not telling you what these interventions are, and I'll get to it later, but you can see this intervention, this gray bar, doesn't peak as high, but it's much more prolonged in the elevation of the oxygen consumption rate. So choosing any single arbitrary point sort of loses some of the context that you gain by being able to longitudinally follow these cells for a long period of time. And a lot of the surprising power we've gotten out of this device is that type of behavior that you would never see in snapshots. So we're playing multiple ways of quantifying this data because each way sort of suggests something different. Yeah. Okay, so and I'll just say that we've kind of employed this strategy, measuring lipid secretion, measuring OCAR increases as we do an intervention under lipid-rich conditions, and have been able to show that certain interventions we've done do indeed seem to promote beta oxidation and decrease lipid secretion. For example, we've come up with a lipophagy inducer that seems to do this and published on that. We have some unpublished data, probably not surprisingly, that if we put in an electron transfer, a uncoupler, mitochondrial uncoupler, that we can titrate the uncoupler to a non-toxic concentration that also does the same thing, decreases the amount of lipid secreted and increases the amount of lipid consumed. Dan and Jim have this paper that's in submission in which they have intervened on this key enzyme here that allows for conversion of acetyl-CoA to malonyl-CoA. And by inhibiting that, they are then feedback inhibiting this inhibition. This inhibition allows for fatty acids to get into the mitochondria for beta oxidation. So if you inhibit the inhibitor, you should be able to release beta oxidation to happen, and their paper shows that, and they also see decreased secretion and deposits in their cell culture models. So having at least a little bit of proof of concept that this idea works and having the tools to work on it, we have a lot of data, and 3 little vignettes have come out of this data that have us somewhat scratching our heads, and I want to present those things mainly for thought-provoking reasons and to get feedback if anyone has it. So the first vignette I'm going to present is that, similar to what Dan mentioned the last time, we're not really sure actually how much beta oxidation is normally happening in the RPE. And let me give you a couple of pieces of data to support that question. So this is in conjunction with Jim and Dan, our lab and Jim and Dan Jim's lab, and in particular with Dan. We fed our RPE cultures with C13 on a palmitate or C13 on glucose. These media were exactly the same otherwise. So it's just a matter of tracing where one label is going versus the other label. As you can see, if we feed 300 micromolar palmitate to our cultures, the palmitate is rapidly rapidly consumed from the supernatant. So it's gone from the supernatant by 13 hours. And if you track how much of a TCA cycle intermediate is made, you can see certainly that the palmitate is contributing to citrate production, more so during this time period than glucose is contributing to citrate production. So that certainly suggests that the RPE is doing some beta oxidation. On the other hand, if we take our cells and we put them, subject them to this recipher device, and under one condition, we have no fatty acids. It's serum-free media, so that we're avoiding any other extraneous fats. And in another condition, we have 300 micromolar palmitate. You can see that the difference in OCAR is pretty modest. If we keep letting those cultures go, we're not changing the media at all. We're just continuously measuring OCAR. You can see as day 4 and then day 5 comes around that the OCARs are dropping because all of the nutrients have been consumed in the media, and there's literally no substrates left for the RPE. If we at this point in time spike in, so we're not changing the media, we're just spiking in PBS or some sort of non-nutritive volume compared to spiking in palmitate with the same volume delivered in the same vehicle, you can see that the palmitate here causes a very large increase in OCAR. So that suggested to us that if the RPE culture is kind of sitting with its happy media, it's not doing much beta oxidation, but it's certainly capable of doing beta oxidation.
One comment, Jason, or question, I'm not really sure on the last slide. It seems like there's a delay in the decrease of OCR over time with fatty acid. Is that right?
Right. So the question is what we think is likely happening is that there isn't an immediate large consumption of the fatty acid, but as other mitochondrial substrates are being depleted and the fatty acids are still around, they are sort of reaching for them to be consumed. It also, an alternative interpretation based on this data is that they are being consumed, but just the rate of consumption, the rate of beta oxidation is not massively large compared to, when they're deprived of other nutrients.
Yeah, putting those two figures together, I was thinking something between those two hypotheses, which is that maybe the fatty acids are favorably consumed and the things that would normally be consumed are, they're waiting until later. But I think it's kind of all possible at this point.
Right. I agree. We don't know. Either one of what you said is possible. I think the main notable thing just is that the amount of spike that we get is just much less sort of initially, than it is if, later on.
Jason,
And,
this is sorry, just a very
ahead,
quick
Karen.
question here.
Yeah.
Very interesting finding. So I'm just wondering, since it's 5-day culture or 4th-day culture, so what is the pH change in the medium, and also how healthy the tissue is without changing the medium?
So these cultures have no depth when we spike in the media here. We long ago, this was like, I don't know, 5, 6, 7 years ago, did do an experiment where we actually changed the media once a week in our cultures, just to see what would happen. And we found no drop in tear or morphology or other health, which is surprising to us given what we're seeing here. But my sense is that right about at 6 to 7 days is where you're, you know, teetering on the edge of everything crashing, but just hasn't quite yet. And I'm pretty sure if we went to 8 or 9 days, you know, everything would have gone down the tubes. But at this point where the OCAR is making a turn and coming down, we can do the recoverability. We can change the media, and then the OCAR will go back to where it, you know, where it would be happy. We can measure the tier, the cells are fine, et cetera.
Thank you.
I think, sure. I think
You have some.
Question for you.
Yeah, go ahead.
What is so the concentration of palmitate that you're using, what is that in relation to the physiological levels of palmitate that RP cells will be, I guess, come into contact with?
The answer is we have no idea. You know, I can't, I do not know what the, you know, physically what the RPE contacts, because there's so much happening, you know, between outer segments and diffusion rates of LDL and HDL, which are the primary, you know, serum-based lipids, plus any free fatty acids that are around. But I can say that in general, for cell culture experiments, especially liver-derived cell culture experiments, you know, which is kind of where the gold standard's been set, these types of concentrations in the 50 to 300, 400 micromolar have been the pretty standard concentrations that are used. And so that's basically why we utilize them.
Is it palmitate conjugated with fatty acid-free BSA, for example, just to increase the solubility, or just a palmitate?
No. These are all, I'm sorry, I didn't, this is all a BSA palmitate conjugations.
Okay, great.
I think Sri,
I guess
Kat,
another-
Martin, Paul, and Kathy all, oh, sorry, go ahead.
Oh, go ahead. Sorry.
Kathy, do you want to go?
Sure, I might have the same question as Shri does. So I'm assuming this is serum-free media. Do you have any sense of the level of canonical autophagy between days 2 and days 5? And have you done this experiment in the presence of an autophagy inhibitor?
Those are great questions that I should have thought about, but had not. But those are great questions. So the answer is no, haven't done it. Would love to think about that for many other, I'm sure the reasons we're both thinking in our head. So yes, great ideas, haven't thought about it. Shri?
So you showed that media depth has an obvious effect on the oxygen consumption rate, and you don't change media here. So I'm assuming you just put the lid on and you're recording continuously. So how do you account for media evaporation, if any? And have you considered using a heated lid, like 40 degrees Celsius?
Yeah, so there's some nice features of this device. I'm not a salesperson for the company, but the device has a humidity detector right on the inside of the lid, so I know what the local humidity right above the cells is at all times. We fill it's a 96 well, but we don't use, there's not cells in all the wells. 2/3 of the plate is filled with water on the wells to keep the local evaporation rates low. And it's actually even better than that. This device, it doesn't just tell you OCAR, it will tell you the oxygen, the actual oxygen concentration. If evaporation is happening, the volume goes down and the oxygen concentration available at that point on the lid will be higher. So you can watch the O2 curves and see that they're steady instead of coming up, to know that you're not having any evaporative effects. The lid is also heated. So all of those are under consideration for the device.
Hey,
Okay, Martin Paul has been very patient. Sorry.
hey, Jason. So at that day 5, when you are spiking in this fatty acid, the cells have been in culture for almost 5 days. You started with 5.5 millimolar glucose. Could it be that also at the same time glucose is already depleted? And what happens should you spike in the same amount of glucose together with the fatty acid? And with regards to your earlier statement that the goal is to promote fatty acid oxidation and allow glucose to the photoreceptors. So should you combine both, what would be your expected outcome?
Right. So I love the questions, and therefore I may not get to the part of the talk that answers your question. I'll jump ahead to basically say that we have data that we think suggests that there are preferential metabolites that the mitochondria, that RPE mitochondria are using ahead of the fatty acids, and that so right now we're in a search for the selective metabolites that are being utilized ahead of or prioritized ahead of the fatty acids. And we were inspired by these types of experiments. So what we've done is we've collected media at various time points during the depletion rate and done metabolite analysis to understand which are depleted. Then we are making up custom media with only that metabolite missing and redoing the beta oxidation experiments to see if we get a much bigger OCAR spike, so that instead of it just being this global loss of all metabolites, which is kind of uninteresting, is there some specific competitive interaction between certain metabolites and fatty acids. I think was that getting to your question, one of your questions, or did I miss your question entirely?
Well, I will wait till I see the rest of the data and perhaps come back.
Okay. So the point of this slide is that certainly the RPE is doing beta oxidation, capable of it. Maybe it's not doing it as much as we thought. That's the main point of the slide. So I wanted to just turn to in vivo, what happens if you eliminate beta oxidation in vivo? And fortunately, we have a lot of genetic data to look at here. There are a lot of mitochondrial beta oxidation disorders in humans, and we can look at the natural history of what happens to those patients. So this disease, which is part of the trifunctional protein complex that's involved in key step of beta oxidation, LCHAD deficiency, definitely has retinal and in particular RPE degeneration as a feature of the disease. And I think that led the field to at least initially believe, okay, RPE beta oxidation is important for RPE and outer retinal health. But if you look at the entire list of known genetic disorders that involve beta oxidation, the only one that causes a retinal degeneration, and in particular RPE degeneration, is indeed this LCHAD. And the mouse model has been built that recapitulates the human disease. However, the other subunit of the trifunctional protein complex does not cause a retinal degeneration. And in fact, the defects in LCHAD, the more mild they are, the more likely there is to be retinal degeneration. The upstream product of LCHAD that builds up because of the enzyme deficiency is hydroxyacylcarnitines. Hydroxyacylcarnitines are exquisitely sensitive to oxidation and therefore promoting oxidative damage in the cell. So Mark Panisi and Melanie Gillingham, OHSU, and now Retinal Foundation of Southwest have a pretty compelling set of data from the patients that it's really this buildup of the toxic metabolite that's causing retinal degeneration in this disease and not the lack of beta oxidation. I will also bring your attention to the CPT1A deficiency, which in humans does not cause a retinopathy. And Dan presented work that Kathy and Nancy and now Jim and Dan are working on, in which the mouse model in which CPT1A is knocked out from the RPE, at least on first glance, it needs to be much better sort of phenotype, but on first glance doesn't have an overt retinal degeneration. So I bring all this up to say, maybe the RPE is doing beta oxidation, but maybe it's not absolutely critical for outer retinal health. And maybe something is inhibiting it from happening at full speed. Certainly there's large mitochondrial capacity in the RPE. Certainly there's a large lipid load. So you would think these two things would lead to a lot of beta oxidation, but maybe some of this data is suggesting that that's just not happening on a regular basis. So as we were searching for reasons that there was less than we anticipated beta oxidation, we really prompted by talking to Jim and Dan, first asked, well, is it just a matter of energy abundance? Maybe in cell culture, you have such an overabundance of nutrients that the RPE just doesn't reach for beta oxidation because it just doesn't need it for energy purposes. But if we put our cells in culture and we let them go long enough that we get to see a good palmitate spike when we spike it in, we can see that the OCAR has not dropped, and therefore we can make the pretty good assumption that energy abundance is equal here as it was, anywhere else along this flat part of the line. And so that suggests that it's not just, an overabundance of energy that's inhibiting beta oxidation, because here there's still energy available, and yet beta oxidation is definitely promoted if we spike in at this later time point as opposed to earlier. So we started just on a candidate-based approach, asking, well, what if we take out one metabolite versus another metabolite? Can we see an increase in beta oxidation in our RPE culture? So here, the up top is the metabolite that we have selectively taken out of the culture, and then we are looking for a beta oxidation spike, and with means we have palmitate in the culture, and without means that we have with means we have glucose in the culture, and without means we have no glucose in the culture, and we're looking at how much palmitate is causing a delta OCAR. So all these cultures are getting palmitate, and we're looking at how much the palmitate increases OCAR, and if glucose had an effect on inhibiting OCAR, then we should see a difference between with and without glucose. We should see a difference between with and without glutamine. We should see a difference between with and without pyruvate. And in all these cases, we didn't see a difference.
What time in culture is this?
This is just right away in culture. Okay. So there's no depletion here. All this media is totally replete. It's just all these media are completely composed, but they are missing this one component to them right now. So, but just to emphasize again, if we wait longer in culture and we spike in palmitate, if we wait 16 hours, we get a certain OCAR spike. If we wait 48 hours, we get an even higher OCAR spike. So what we've settled on doing now, and just to make the point that there's lots of other data out there, including from Kathy and Nancy and others, that have shown that you definitely can see an OCAR spike when you add in fatty acids in a minimal media, like a Ringer's type solution with just some glucose around, for example. So all of that, again, suggests to us that something needs to be depleted in order to see a lot of beta oxidation. And our first initial guesses, glucose, glutamine, and pyruvate, were not correct. So we're now basically, as I said, collecting media at various time points after addition to the culture and seeing which metabolites are most depleted, and then taking those metabolites and creating media that's devoid of just that metabolite and repeating these same experiments until we hope we find something that does really compete with beta oxidation.
So Jason, this is just shotgun metabolomics then?
We're doing right now, we're starting our focus with amino acids. So we're doing all 20 amino acids because there's, of course, decent data that certain amino acids are highly consumed by the RPE. If we don't see anything with amino acids, then we'll expand to sort of more a larger metabolomics panel.
Okay.
Okay, so I got through vignette number 1, and we'll get through whatever there is for the hour. The second.
Jason, before you go on, I'm sorry. You didn't tell us what the millimolar concentration was of those additions. Are they 10 millimolar?
Oh, yeah. So these are all very standard additions. So the glucose is at 5.5, a physiologic 5.5 millimolar. The glutamine is at 2 millimolar, which is both standard in cell culture and just slightly above, known serum levels. And the pyruvate is at, I forgot if we did 1 millimolar or 500 micromolar, both of which are pretty standard for cell culture and slightly above, above what would be seen in serum.
And the molar concentration in the palmitate?
The palmitate was all these are at 300 micromolar.
Okay, thanks.
Yeah. Okay, so each of these vignettes kind of links to the other, but the second question was, okay, we're doing all these OCAR experiments. We've chosen palmitate because it's standard, but what happens when we put in other fatty acids into our OCAR system? What happens to their OCAR rates, and what happens to their ability to promote lipid secretion in the RPE? And so do all the fatty acids that the RPE sees equally contribute to this push and pull between lipid degradation and lipid secretion, or do some tend to favor degradation and some tend to favor secretion? So this is John Hahn's work, and I'll just sort of show this experiment right off the bat. Over on the y-axis, we have again the delta OCAR. Keep in mind again that we initially always see this kind of plateauing as the oxygen gradients are being set up in our culture system, and we have a variety of different fatty acids here. So on the bottom here is just the media change. So we had the cells plotting along over before you see the graph, and then we at 1 day we just changed the whole media, and this one just got another media change, and it stayed at its sort of standard OCAR that it was at before. If we put in palmitate, we saw there's some depletion of, if we put the palmitate into depleted media, we see this palmitate spike. But if we put in oleic acid, we see a less of a spike. And if we put in these fatty acids that are destined for the peroxisome, so erucic acid is 22,1, otherwise known as Lorenzo's oil, thought to be pretty highly specific for peroxisomal targeting, a major component of rapeseed oil. If we put in erucic acid, or of course our favorite polyunsaturated fatty acid, DHA, we see some increase in OCAR, not as much as oleate, and certainly not as much as palmitate. These are all being put in at equal millimolar. I can't remember. We might have done 100 or 200 here. I forgot because we wanted to, because in general the 100 was more standard for the types of concentrations being given for erucic and DHA, and we wanted to keep them all molar equivalent. So there seems to be a difference in how much each one is promoting beta oxidation. What about on the secretion side? And these experiments are just literally baby experiments early on, so all of this could change. But it seems fairly consistent that oleic acid, so we're looking at ApoE secretion again as a proxy for a lipid secretion. we've established that as more lipids get secreted, more ApoE gets secreted. And so if you look at ApoE secretion under oleic acid treated conditions, you see more than, for example, palmitate secreted conditions, or when there's no fatty acid around. And the Arushik and DHA seem to be less than oleic, but more than palmitate. And at least for oleic and palmitate, we've done enough of these experiments to feel pretty certain that oleic consistently causes more ApoE secretion than palmitic acid. I have to say, it doesn't feel this dramatic to me normally, but the quantification came out this way, that here's palmitate over here in terms of the amount of ApoE secreted, which is basically the same as when you put in just serum-free media. And when you put in oleic acid, you get a lot more ApoE secretion, pretty comparable to if you have just serum around that amount of ApoE secretion. So this led us to kind of wonder, as a pathway, we've drawn this little diagram here. The RPE, of course, faces lots of different lipid loads. We've been a little reductionist right now about just feeding it fatty acids. It normally, of course, sees outer segments and lipoprotein particles, but it does see some fatty acids, especially medium chain fatty acids that are probably delivered to it directly from the serum. And of course, we think that at least most of the LDL and HDL comes in through receptor-mediated endocytosis, which means it gets into the lysosome. Once it's released from the lysosome, it ends up in the ER. From the ER, we know that lipid droplets temporarily seem to form in the RPE, but maybe each fatty acid really has a different preference. For example, oleic acid may form more lipid droplets and be more shuttled towards secretion via the ER, whereas palmitate may be a great substrate for mitochondrial beta oxidation and makes its way into the mitochondria. And of course, Arushik and DHA, we think, and this is what we need to be proving, will make their way into the peroxisome and then be released as shorter chain fatty acids that then can make their way into the mitochondria. And so, each one of these, by being handled differently, really may be promoting secretion versus degradation at a different level. In particular, we've got this sort of simple experiment, which we definitely need to confirm and maybe, totally contradicted in follow-up experiments. But this simple experiment in which oleic acid both has less OCAR increase but more lipid secretion increase compared to palmitic acid made us wonder about this enzyme, a sterol desaturase, that allows for the interconversion of unsaturated and saturated fatty acids. This has been studied in the RPE before, but we're wondering whether or not the inhibition of this enzyme, which would potentially promote more retention of palmitic acid as palmitic acid, could promote beta oxidation and therefore decrease the amount of lipid secretion to drusen and reticulosuttodrusen. So that's down the line, but it made this target of some interest to us.
Hey, Jason. How many of the peroxisome biogenesis disorders have a retinal degeneration phenotype?
That is 1 or 2 slides from now.
Okay.
And the answer is a lot. It's unlike mitochondrial beta oxidation disorders, peroxisomal disorders really hit the retina hard.
But not all of them, right?
Not all of them, but
Yeah.
a lot of them. Yeah.
Okay.
So I probably won't be able to really talk about this vignette because we're running up on time, but I'll at least just introduce the idea that maybe, and this is not our idea for sure. Kathy introduced this in some of her previous papers that have studied peroxisomal biology in the RPE, and many others have mentioned this before. But once it starts happening to you, you start realizing what everyone's talking about. So we are wondering how much mitochondrial peroxisomal crosstalk there is happening in the RPE, especially in aged RPE as the mitochondria is compromised and perhaps more lipids that should have been destined for the mitochondria get destined for the peroxisome instead. So just as a brief reminder, peroxisomal beta oxidation very much parallels the concept and idea of mitochondrial beta oxidation, which I have on the right side here. But there are some key differences. Of course, the peroxisomes, the idea is that the longer chain fatty acids get into the peroxisome. In the mitochondria, the canonical teaching is that it's 18 carbons and fewer that get into the mitochondria. In order to get into the mitochondria, you have to conjugate carnitine to the fatty acid. That is not canonically true in the peroxisome. The first step in the peroxisome involves the conversion of O2 to hydrogen peroxide, which is why the peroxisome also expresses such high levels of catalase to detoxify the peroxide that's generated during the process. But the rest of these steps really are, I mean, they're different genes and proteins involved, but they really do a similar conceptual thing. I'm going to skip a figure I totally ganked from Mike Landowski, except just to remind you that besides degrading fatty acids in the peroxisome, DHA has to be synthesized via a step in which it enters the peroxisome as THA. Two carbons are removed through peroxisomal beta oxidation, and that allows for the eventual synthesis of DHA. So how important are the peroxisomes in the RPE? There's a whole range of peroxisomal disorders, and many of them prominently feature RPE degeneration. So I've taken a Zellweger syndrome patient here. I can't remember which PEX was defective in this patient, but a really nice review of the peroxisomal disorders and their ability to cause retinal degeneration just came out in Ophthalmology in 2023 with Braverman as senior author, if anyone's interested. You know that this is an RPE-based degeneration because of all of the pigment disruption. Plus, on the OCT, the clinicians recognize this, but when the RPE is gone, you get OCT signal bouncing off the choroid, and so the choroid looks extremely reflective here, very bright, which is not normal, and that's because the RPE is no longer blocking the OCT laser beam from reflecting off the choroid. It would normally, the RPE would reflect that light coming back to the detector, and instead it's now bouncing off the choroid. So we know that peroxisomal disorders feature prominently RPE degeneration, unlike mitochondrial beta oxidation disorders. Certainly, peroxisomal activity varies diurnally with outer segment phagocytosis, and this is a paper that Kathy's group published in 2019, in which they showed, just take it for right now, that the y-axis is showing some degree of peroxisomal activity, beta oxidation activity, and you can see that there's an increase during the transition from lights off to lights on when we know that outer segment phagocytosis is happening. You can see that there's this increase in peroxisomal beta oxidation activity that then goes back, back to sort of some basal rate once outer segments have been phagocytosed. And recently, Miriam Bays, who's at KU Leuven in Europe, and her grad student Sai, showed that if you knock out the key enzyme, one of the key enzymes in peroxisomal beta oxidation, you get a very rapid RPE vacuolization and degeneration. So here's the control, here's the knockout. And this degeneration is happening over the course of 30 to 60 days. If you cross it with a rapid degen mutant, in which basically there's no outer segment shedding, because by the time the outer segments are mature, the photoreceptors have basically died. If you cross the mouse with that RET-degen mutant, you rescue the RPE phenotype, strongly suggesting that the toxicity is coming from outer segments with their VLC PUFAs and DHA not being able to be handled by the RPE, and then some sort of toxicity arising from all this. All of this to say that clearly peroxisomes are important, and we stumbled onto the potential of the peroxisome to be a shunt when the mitochondria are inhibited, because we started doing these experiments in which we inhibited mitochondrial beta oxidation by using the drug atomoxir, which, as people know, helps to prevent the carnitine conjugation to the fatty acid, which allows entry into the mitochondria. In the process of adding atomoxir, oh, and I should say that Dan in Jim's lab had well shown that very low doses of atomoxir in the low micromolar range really totally abrogate the conversion of C13 palmitate to a product, beta hydroxybutyrate, that has to be made through beta oxidation. So we know that at the lowest concentrations of atomoxir, we should be abrogating mitochondrial beta oxidation. And yet, when we put our atomoxir on our cultures, so on the y-axis here, you see this is the delta OCAR, and you can see here we just have media, and here, because we deliver our palmitate with BSA, and we wanted to make sure the BSA alone wasn't contributing to any OCAR spike, we are just delivering the BSA, which doesn't cause a meaningful increase in OCAR. If we deliver palmitate by itself in these depleted media, we get a spike. And if we add even more than 4 micromolar, 40 micromolar of atomoxir, we've tried it with multiple concentrations, we get less of a spike than with palmitate alone, but then it's every time totally consistent, without fail, we get this long tail in which the OCAR stays elevated for a longer period of time. Now, there's lots of possibilities here. One is atomoxir is wearing off, and we're going to try to address that by repeatedly spiking atomoxir in. But another possibility is that atomoxir is not completely inhibiting peroxisomal beta oxidation, which is controversial about whether it does or not, and we are shunting some of our mitochondrially destined beta oxidation material, palmitate, into peroxisomes instead. So we're really trying now to understand whether this is true, and further, whether or not peroxisomes act as a sort of escape valve for dysfunctional mitochondria to allow for handling of not just VLC PUFAs and DHA, but also fatty acid substrates that are classically thought to really be the confines of the mitochondria, like palmitate, for example, and oleate. So I know I'm over time, or just at time, and so I'm going to skip these last couple of slides and just get to the conclusion. Our conclusions then are that beta oxidation may not be happening spontaneously in the RPE as much as we originally supposed, and I presented some in vitro and then some corresponding genetic in vivo data to support that idea. We think that other metabolites may be tonically inhibiting beta oxidation, and we're trying to search for what those metabolites are. We have previously shown, and we continue to think, that even though there is tonic inhibition of beta oxidation, if we can push past that tonic inhibition, either pharmacologically or by manipulating metabolites that are competing with beta oxidation, we may be able to therapeutically decrease the secretion of lipids that contribute to drusen and RPD, while promoting the energetically, while promoting fatty acid degradation, which provides energy to the outer retina. We clearly have shown that different fatty acids seem to lie at different points along the spectrum of promoting mitochondrial beta oxidation, versus being secreted. And we're interested in pushing the interconversion of these fatty acids such that we get more of the fatty acids that are able to be degraded and less secreted. And then lastly, reduced mitochondrial activity, as in AMD, may result in a shunting of things that normally would have fatty acids that would have normally gone to the mitochondria into peroxisomal beta oxidation, the long chain and medium chain fatty acids as escape valve. And if mitochondria are dysfunctional in the RPE, this is definitely not a revolutionary idea. It's been mentioned many times. Can the peroxisome act as an escape valve, and can we promote its health in AMD? Either transcriptionally upregulating it, for example, with PPARs, or promoting autophagic turnover, which again was an idea that Kathy had brought up before through pexophagy, et cetera. So I'd just like to acknowledge the people that did this work that I presented here. Jillian is the co-lab manager for the lab. Chitao is now a research investigator in the lab, and unfortunately just fractured his ankle and is just recovering from surgery right now. John is the postdoc that was generously loaned to our lab from Nancy Philp, and he's currently in Korea at 1 a.m. in the morning listening. Dan and Jim for this collaboration that we continue to have with them, and Nancy for continuing to provide mentorship and advice. And then the rest of the lab members, Keisha, Aaron, and Caitlin. Keisha's our lab manager, Aaron's a senior scientist, and Caitlin's our graduate student. And then our funding, both from the NEI, from the Grossfeld Initiative that we have here at the University of Michigan, and then some other foundational awards. Thank you very much.
Nicely done, Jason. Obviously, I'm going to be grilling you at our next committee meeting for your Kate Award. But one thing I'm interested in, because there was, I think, a third or fourth point down, testing this therapeutic, I assume you're going to be using an animal model to test that. So what's your choice of AMD-like animal?
This is the perennial question. I can see Claudio has his hands up. I'm sure he's going to tell me we should be doing it in the TSC, the TSC one knockout, which, by the way, my colleague Tom Woobin has here, and Eric Way, who's on the call, is working on as well. You know, to me, the model would be most helpful is the model that I believe has deposits coming from the RPE. So if the model is hyperlipidemic in the bloodstream, I'm worried, because no matter how much I modulate what's going on in the RPE, all that lipid from the bloodstream is just going to deposit down on Brooks. At the same time, you know, there's not that many models that build up significant deposits in mouse models that build up significant deposits. So I'm certainly playing around with it. I can tell you I've thought about the TSC1 model. I've thought about Goldis's LXR alpha model. I've thought about the CLIC4 model that just recently came out. I've been in contact with each one of these researchers, and I'm kind of just weighing it right now. In addition, even though there is no phenotype for the CPT1A mouse, I am curious if it gets stressed and aged, whether deposits would build. And so that's also of strong interest, and we're thinking about taking on that project. So I dodged your question entirely, Steve.
What I wanted to see, actually, more importantly, your thought process, which is encouraging that you have considered this range. And I was very happy to hear that you didn't consider, obviously, a lipid-loading model like a high-fat diet or ApoE null or any of these other things that I don't believe in. But at any rate, those are some reasonable options. You're just going to have to, there's no good way to a priori know which one is the best or better. So I think you're just going to have to empirically go down the line.
I agree. Slog it out.
Exactly. It's great talk. Thank you so much.
I'll start because I raised
Dan,
my hand
yeah,
first,
and Dan, why
expecting
don't you
that.
mediate? Why don't you mediate questions too? So because I can't quite see who's
Oh God,
answering.
I didn't track. There was just a flood of hands that came up simultaneously. So I will just go from, after me, I will go from the top of my little Zoom bar to the bottom, which I'm sorry if you kind of are getting skipped, but or not getting to answer first, asked first. I'll ask first though. Okay, so outer segments, obviously a combination of saturated and unsaturated and polyunsaturated and all sorts of fatty acids. Have you tried this depletion, then outer segment feeding instead of palmitate feeding? And I mean, if not, probably you'd expect it to look like what?
Right. So John is eagerly he's probably asleep by now, because of how late it is in Korea. But John is eagerly wanting to do this, and in fact, we started doing these, basically feeding outer segments and tracking OCAR. But we realized when we ran into this, so we wanted our controls to be atomoxir, and we assumed when we put on atomoxir, we'd see no increase in OCAR with anything. And then we ran into this issue of what you just saw, these very reproducible curves in which the OCAR doesn't go up as much as without atomoxir, but then prolongs way afterwards. And so we are trying to really understand, before we start making conclusions about anything, we're trying to understand what that means and what that is. And including, there are some other tools to potentially more specifically inhibit just the mitochondria. CPT2 has a small molecule inhibitor called L or R aminocarnitine. And we're looking into ordering that and utilizing that. We may ultimately have to go to a genetic, system where we, don't have to rely on small molecules, so we can be sure, that whatever CPT1A is out or CPT2 is out. So we're stalled on the answer to your question because we ran into this confusing thing. But ultimately, that is what we're gonna ask that question, or hopefully answer it. How much does outer segments versus palmitate, increase OCAR?
Yeah. I mean, I guess my expectation would be somewhere in the middle, right? As a mix of fatty acids.
I can tell you the initial data that has do not hang any bit of weight on it, was exactly what you just said, that outer segments do not promote OCAR as highly as palmitate it does, just like you said. Yeah.
Okay. I'm going to also ask the question I had in the chat, which is, when you saw that the other fatty acids like oleate and some of these unsaturated fatty acids didn't have as high a raise in OCAR, did that have a prolonged increase in OCAR relative to palmitate?
No, not.
Okay.
So let me rephrase that. The oleate did not. The peroxisomally derived ones may. We have to sort of piece that apart. That was right here. So oleate is here, and it's hard to see, but it comes down like this one. The peroxisomal ones, we can't tell. We may have to do them again. I can't tell whether they sort of extend out much more. Probably not. But they all. It's this one here, DHA. You can see peeks through at the end here, not of any significance, but we need to repeat these to know whether or not that one is indeed lasts longer. Like, again, that maybe peroxisomal beta oxidation is a slower process because it ultimately has to utilize both the peroxisome and the mitochondria, because the downstream products of the peroxisome have to make their way into the mitochondria. And if they don't, we presume that will cause feedback inhibition and slow down peroxisomal beta oxidation.
Okay. I've already taken more time for my own questions than I have a right to, so let's go to Kathy, who's next on my screen.
So Jason, really, really nice talk. Just two thoughts. And I'm sure you know this, oleic acid is actually the preferential lipid for lipid droplet formation from the liver literature. So as you said, it's not particularly unexpected to see what you're seeing here. The second thought is we just published on a knockout that some of you heard about it, RD, on MTP specific knockout of the RPE, which decreases or completely almost eliminates beta lipoprotein secretion that is RPE specific. The reason I'm bringing it up is because in that animal model, we see a lot of subretinal type deposits that are full of unesterified cholesterol. So if that is something you are interested in thinking about in some way, just let me know. And it's in FastSub Journal about 2 weeks ago.
Okay,
So
great.
really a question, more a thought.
Great, thank you. I didn't realize that was published now, so that's good. I'll take a look at that. But in particular, I also did not realize that there's these subretinal deposits, which would be great to look at.
Mike?
Yeah, I had a question and then a comment. I like all of the hypothesis between the interaction between peroxisomes and mitochondria. I mean, a lot of our work is supporting that as well. So I'm not surprised that that will turn out to be true. My question for you, it actually is like one of your earlier slides when you talk about the palmitate oxidation, how it's like when you add it to your culture, you have a little bit of an increase versus the other one. So I was wondering, have you considered looking at the activity of SCD1 to see if the palmitate that you're adding, that you're having a portion of that pool actually becoming
Unsaturated.
Yeah.
Yeah.
Because
so
as you
yeah. So I brought up this little sort of thing here when I mentioned, oleate versus palmitate, and that we're very interested in SCD1 in particular. Like, can we manipulate it? It's highly expressed in the RPE. Would it change the balance between saturated and unsaturated? But your question is it actually happening? And I can tell you the data, I have not analyzed the data fully, but we did do we put in C13 palmitate into our cultures and then did a shotgun untargeted lipidomic analysis of where the C13 label ended up. And I definitely saw the C13 label end up in species that I would identify as oleic acid. So I strongly presume SCD1 has to be quite active, in order to have seen that. And the oleic acid species were mainly in tags and phospholipids, where the C13 label ended up. So I think the answer is yes, I'm 99% sure that it's actually happening.
Yeah, because that's a good target. I mean, we see some interesting things happening with SCD1 as well, but it's also actually relevant, especially when you talk about peroxisomes, is that upregulation of SCD1 can alleviate a lot of the defects in peroxisome defect models. And that's believed to be because you're essentially blunting the formation of these toxic lipids that can form in those cells. So it's nice to see that that's something that's on your radar, because I think that actually is going to pan out to be something that's really important.
I'll chat with you after, Mike. Thanks.
All right, sounds good. Good talk.
Great.
Jason, I was wondering how exactly RP is handling all these exogenously supplied free fatty acids. So obviously it's very hard to track, but maybe there's a way. I was just thinking about it. And maybe you can use HA tag BSA to load your free fatty acids, and then you track HA if it's being trafficked to lysosomes or ER or to mitochondria or peroxisomes. If it's going to lysosomes, you would lose your HA signal.
You're saying by just HA?
Yeah, just buy commercially available BSA, HA tagged BSA, and conjugate your fatty acids to HA tagged BSA and track HA.
Yeah, I mean, I think if our bandwidth is of course limited, but I would say if I want to do that, which I've certainly thought about, they're not natural, but they're the Bodipi, C12 Bodipis that at least vaguely, are supposed to mimic in chain length, C16, C18 or so. The other issue with BSA tagged fatty acids is it's of course hasn't actually been well worked out exactly how the fatty acids get into the cell. And there's a hypothesis that the BSA doesn't itself get endocytosed or pinocytosed or anything else. It's just, it creates a high local concentration at the membrane, and then there's a slip off of the fatty acid right through sort of the membrane without the BSA coming with it. That's certainly, I've seen plenty of data to support that that's a possibility.
How
So
would that tie into classical endocytosis then?
I'm sorry?
How would that be endocytic uptake if it's being just...
No, that's what I'm saying. There's
going
at
straight in
least
the
some
membrane.
amount of fatty acids that literally just passively traverse the membrane, and the purpose of the BSA was not as a delivery into the cell. It was to keep a highly hydrophobic thing at a high enough concentration next to the cell surface to allow for passive efflux of the fatty acid off the BSA and into the cell. There's been some work to support that idea.
The other thing is, I know you showed that oleic acid goes into probably lipid droplets, or at least it triggers lipid droplet formation. And you see a difference between oxygen consumption rates between oleate and palmitate. So maybe it would be really interesting to also do this experiment, compare oleate and palmitate in the presence or absence of lipid droplet formation. There are commercially available inhibitors.
I'm in total agreement. Your former boss is a mentor on my KO8 committee that is exactly trying to do that. We're trying to manipulate lipid droplets, which turns out to be harder than we thought, and see what the consequences are of manipulating when different lipid loads come in. So I totally agree with you.
Thank you for your patience, Claudio.
Yeah, sorry.
Thank you, Jason. A question and / comment. Comment to the TSC1 rod. If you want to use it, you can speed it up by just putting them on a high-fat diet, and within 6 months you will see uniform lipid accumulation at the RPE. So that would make it more feasible. And they develop also subretinal deposits. We just never characterize them. Just as a comment, if you want to use the raw TSC1. And then I had a question to your amino acid depletion. Have you gotten already to leucine, arginine, and methionine?
Yeah, so we just finished the mass spec, like, the serial mass spec, and I'm analyzing data. So I'm definitely interested in arginine and methionine, but have not tested them yet. But they are on the list of things to test.
Yeah, because all 3 amino acids regulate mTOR, and mTOR inhibits also, or well, mTOR inhibition promotes beta oxidation. And it may be, especially for the RPE, the level of amino acids may be a readout of phagocytic activity, because, it's full of protein, basically, right? It's lipid and protein. So it may be one way it is regulated, and those 3 amino acids may be very critical in that regulation.
Yeah, it's a good suggestion. And indeed, we're definitely aware and thinking about whether or not amino acids could be directly competing or could be competing via mTOR signaling, which would downregulate, beta oxidation. I think Roxana.
Hi, Jason.
Hello.
That was excellent presentation. Really fascinating the way you model in the culture and measure all this sort of oxidative processes of the lipids. I have a question. Do you know does light play a role in the fate of lipid metabolism in the RPE?
A light, you mean like LIGHT? Well, we're, I think, poorly equipped to directly study that because, you know, we manipulate cultures and have to see what we're doing. But I mean, this is just my, I don't think this is my personal opinion. I think this is a lot of people's opinions, perhaps yours as well. You know, it's so contradictory that we have massively oxidatable lipids sitting perfectly exposed to light. It's like, where else in the body would that like terrible confluence happen? And so, you know, perhaps you're getting at the point that, you know, if you have lipid peroxidative products, are they actually being handled in the same way as non-peroxidated products? And I certainly am very interested in that idea, and in you know, also the links to ferroptosis and signaling as well. We just haven't gotten to that, but that is, you know, I think ultimately that's definitely testable, and I have on my long to-do list somewhere that, you know, that basically to sort of oxidize various types of lipids and understand whether their behavior in the culture system changes, you know, based upon on that pre-oxidation.
All along about this circadian rhythm, and particularly this ball of the engulf outer segment, that peak apparently it is, you know, at certain time, but the digestion of proteins and possible these lipids also are, you know, different at different time of the day.
Yeah, you mean they're delayed, you mean they're lagged from the, yeah.
And likely modified during that time of the day. And how is this sort of handle giving the different mutations? That will be interesting to look
I
at.
assume you also probably believe that the ABCA4's RPE function
100%.
has to do with some of what you're talking about here.
Loads of data. So
Yeah.
preparing this manuscript, because yes, from the mechanistic standpoint, it's quite mind-boggling what happens with the lipids.
Yeah.
Particularly for Stargardt. The only difference, so this is where I kind of enlightened my mind with all this sort of secretion and deposits. Well, in Stargardt, it's all inside the cell. It doesn't get to create the subretinal or drusona. It's absolutely just cell handling the lipids, that it's completely different than most of the other macular degeneration are. So that makes it more, you know, kind of standalone.
Right, and uniquely susceptible. There's just no escape. There's no escape. Yeah.
Hopefully this will come out soon. Thank you. It was awesome.
Thank you.
So we have a few questions in the chat, starting with Kira's at 9/11.
Okay. I feel like I've kept everyone, so I don't want to say.
Yeah, maybe if you want, you can. I'll send you the transcript from all this. And
yeah, I can send an email to folks.
Email.
That way, I don't want to keep people, you know, longer.
I think at least
yeah,
some of the questions were answered.
Yeah, great, thanks. And if you just text me the chat, then I'll send an email to people if they're for follow up. Thanks, everyone.